gene acts by itself in determining an individual's phenotype. Rather, it is the result of highly complex and integrated molecular reactions that are under direct gene control, is known as non-allelic or intergenetic interaction. Most morphological traits like height and color are affected by multiple genes. It is the genetic analysis that identify the genes which interact for the particular biological property. Classification is that two interacting genes produce modified dihybrid ratios. Number one, non-epistatic interaction, in which two or more genes control the same general character. Examples are typical dihybrid ratio, which gives 9:3:3:1 ratio, same as that of Mendelian dihybrid ratio, and polymeric gene action, which produces 9:6:1 ratio. Number two, epistatic interaction, in which one gene masks the expression of the other and changes the phenotype. Epistasis is of several types. Number one, dominant epistasis, in which one dominant gene hides the phenotype of the other gene, and F2 cross produces 12:3:1 ratio. Number two, recessive epistasis, which produces 9:3:4 ratio in the F2 generation. Number third, duplicate dominant epistasis, in which the dominant allele of both the gene loci produce the same phenotype without cumulative effect, and the Mendelian ratio is modified into a 15:1 ratio. Number four, duplicate recessive epistasis, in which both genes are required to produce a particular phenotype and produces 9:7 F2 ratio. Number five, that is dominant and recessive epistasis, in which one dominant gene produces the concerned phenotype. Now let us discuss epistatic interactions. Epistasis is concerned, it is the interaction between different genes, that is non-allelic, while as dominance is the interaction between different alleles of the same gene. The first type of epistasis, that is dominant epistasis, as already told, occurs when a dominant allele at one genetic locus masks the expression of the alleles of a second locus. For example, inheritance of fruit color in summer squash. Here, the dominant allele W results in white fruit color, regardless of the phenotype at the second locus, Y. In the absence of dominant W allele, dominant Y results in yellow color, while little y results in green color. Therefore, if two white-colored double heterozygotes are crossed, an interesting phenotypic ratio occurs because of this type of epistasis. Of the 16 offspring, 12 are white, 3 are yellow, while 1 is green. Ratio of 12:3:1 occurs. Q is converted to a green intermediate and then to a yellow end product. It is suggested that the dominant Y allele is required to convert green intermediate to yellow, and dominant W allele inhibits white to green conversion, resulting in white fruit.